RAN peptides are a feature of repeat expansion neurodegenerative disorders, such as Huntington's disease, amyotrophic lateral sclerosis, and frontotemporal dementia. These protocols provide an important standardized approach for quantifying RAN peptide toxicity in the C. elegans model system.
The techniques described are simple to learn and inexpensive, can be performed quickly, and leverage the reproducible features of the C. elegans system such as movement and reproduction. When attempting this protocol, the major challenge is obtaining consistent RAN peptide toxicity and expression. The major factors to control include growth temperature, timing of temperature shifts, and preparation of assay plates.
It is important for experimenters to consistently classify the phenotypes of the animals in this age-dependent process assay. Visual demonstration of this technique shows their classification criteria.
Begin by pouring standard nematode growth medium with 1 millimolar IPTG and 25 micrograms per milliliter carbenicillin into 24 well plates. Streak out RNAi feeding clones in HT115 bacteria onto LB carbenicillin agar and grow them at 37 degrees Celsius for 24 hours. On the next day, pick a single colony into 1 milliliter of LB carbenicillin liquid media and grow the bacteria for 18 hours at 37 degrees Celsius while shaking at 250 RPM. Spot 4 individual wells of the NGM RNAi 24-well plate with 20 microliters of overnight bacterial cultures as described in the text manuscript, and allow RNAi bacteria